Thank you, Alan. I did not remember that. Um, so Benchling is just a web application um, used by hundreds of thousands of biologists across the world. It's a really good software for pretty much a lot of your biology needs. Whether it's protocol analysis, taking notes, a notebook, plasmid design, primers design, digesting digesting design, so on and so forth. Uh, I implore you to try to explore as much as you can with this, and more than I have even, to really get a good grasp of this, because it's going to help you learn a lot of techniques in biology, but also, you know, feel what it's like to actually be like working in a wet lab. Because before you do any wet lab work, you're probably going to be doing some designing to make sure your experiments go well. And so you're going to use benchling for that. And so cool. So today we're going to be talking about a very specific thing, a plasmid. So my first question to chat, anyone can answer, except BIOME members, they can't answer. Or if you're an iGEM, unless you're like not in Stanford iGEM, then you're cool to answer. What is a plasmid? What's a plasmid? And even if you're feeling brave enough, which I know we got some brave people in chat today, what's it used for? Like why are plasmids so cool? So feel free to answer as I get ready to explain that. It is a circular piece of DNA. You're correct. Yeah. Yeah, that's what it is. It's a circular piece of DNA. Here's a good example of what a plasmid looks like. Look at that. That's a plasmid. Uh, yeah, exactly. Transferred, inserted in other bacteria. Yeah. And we call that transformation. So Optimus Prime style, we're trying to transform these bacteria into beasts, to do things, to do our biddings, do crazy things. A good example would be if you've ever heard of one of the big leaders who created iGEM and helped create BIOME, Drew Endy, his wife, I forget her name. I'm sorry. I've only had him in class.
Christina.
Um, what's her name? Christina?
Christina
Yeah,
Smokey.
she's awesome. She made yeast, which are kind of, yeah, uh, create opium using that technique, which is nuts. And don't worry, Drew Endy proved you can't do it at home. So don't go try to set up your opium factory in your backyard quite yet. Um, yeah, good answer. Yeah, that's correct. Cool. So yeah, it's really exciting, right? There's so many powerful things you can do with that. And so we're going to talk about one specific use case that's used all the time. And maybe it's not as powerful as creating opium. However, it's very good. Yeah. If you're here, this is high school engagement, even if you're undergrad, so on and so forth. This is the BIOME webinar. We're here to sorry, there's a question. Just teach you about every week we're going to talk about something different. And this week it's about benchling. So this tool, next week, I think it's about PyMol, but Alan will lead that one. And we have previous weeks too. You can check out our website to find those also, or our YouTube channel. So this week we're going to talk about plasmids and specifically in benchling. Biome.bio is our website. Yeah, cool. So yeah, so this is for high schoolers. I mean, if you're looking to join also our mentorship program, this is going to be a software you're going to use most likely. So it's good to be here too. Awesome. Where was it? Yeah, so those are plasmids. Very cool. So let's start. So we're going to be talking about GFP. So GFP is green fluorescent protein. It's actually, see this little whatchamacallit, the jellyfish. That's where the Benchling got its kind of emblem for, was because GFP was sourced from jellyfish. They fluoresce in the dark. And if you remember from previous webinars, or if you've ever done anything in the lab, fluorescence is used all the time to tag things, to look at how much of something you have. It's really cool. So you want things to fluoresce. However, today we're going to be looking at yeast. We're going to be specifically looking at baker's yeast. The Latin name I cannot pronounce, but if you look at baker's yeast, you can find it and try that tongue twister out for yourself. And we're going to be looking to transform baker's yeast in order to produce this green fluorescent protein. Now, why would we want to do that? Because it's cool. And that's kind of it. We want green bread. Why do you want green bread or maybe even green beer if you're old enough, which I quite am. In October, I'll be making green beer. It's awesome. That's the power of biology. You know, why not push it to the limits and try something new with the knowledge you have? Green bread and ham vibes. Yeah, we can maybe put E. coli all over our ham and make it green too, if you if you want to. Cool. Yeah, awesome. So let's get started. So I'm going to introduce you to first this. This is our first plasmid. It looks pretty complicated, but what it is pretty much we want to find a basic backbone. So a basic backbone is going to have certain qualities. If there's any questions in chat, I'm going to close it just so I can clear my screen up to talk. But if Alan or Lauren, you can interrupt me and just like say, hey, Colin, answer this question or something like that. Thanks. Cool. So we want this basic backbone. And so there's going to be certain parts of this backbone that we're going to look for. And as I look through them, I'm going to kind of tell you what's important. So I already have a couple here that I think are really cool. But I didn't just make these myself. Similar to if you've ever coded a project before, you're not going to just code everything from scratch every time. Or let's say you're doing math. You're not just going to do math from scratch. You don't have to invent Pythagorean's theorem every time you enter a math class. You have a library, right? You have things that are known before that you're going to use all the time. So you want a backbone that has certain aspects to it that are useful to you. Benchling doesn't, you can upload them to Benchling. They don't necessarily have them, but a good website and a website you're going to want to check out, and you can type it in as you work along with me, maybe you can type it in. It's called Agi. You can tell it's pretty much meant for plasmids because look at that. That's just a plasmid definitionally. So I'm at this part here where it's called empty backbones. So why do I want an empty backbone? Because I want to insert my green fluorescent protein into it eventually. So they have all kinds of backbones. You got mammalian, if you transform bacteria, yeast, fly, worm, insect. I don't even know what that is, but cool. So let's look at these backbones. So we got a couple here, so a couple vectors for us. Don't worry about the names. People name them all kinds of things. They're usually named about what they produce and things like that, but don't worry too much about them. Let's just look at them. So first, we're going to look at this one. Ooh, this looks like an all right backbone. So specifically, this is meant for yeast. And how can you tell it's meant for yeast? And why is it meant for yeast? So the first thing I want us to look at are these ORI sites. Now, ORI is just short for origin. So this is the origin of replication. So why do you think you have, so replication, right? You want this plasmid to be repeated over and over again. You don't want just one plasmid in our source. So for example, right, you create your replication fork, so on and so forth at these sites. And that's where the plasmid would just get looped around really quickly and replicated a ton. So you get a lot of this DNA because you want a lot of this DNA. These aren't really super cheap. They're maybe the full plasmid could cost $700 to $1,000. You want this to replicate. So my question for you is, why do we have 2 origin points, right? If this is meant for yeast, why not just have 1 origin point? So if you got answers for that one, I'll now be looking at you. I am an old man. Even my beard's starting to gray. Oh. Twice the speed, yeah, that's not a bad answer. Let's think of some other ones too. We got any other ideas in here? Maybe why 2 origin points? It's kind of a trick question because I didn't give you all the puzzle pieces to put it together. So I apologize for that. I'm being kind of a meanie with my question, but it is something to think about and something that's super important to make sure you have in your plasmid, depending on what you're doing. So I'll give you like maybe a minute more for cool answers and stuff like that. Double-stranded. Good guess. 2's better than 1. That's wrong, actually. Second place, they got like second. I'm forgetting the rhyme. Third is the worst. Second is no. What's that rhyme?
Second is the best.
No, it's not. Wait, is it?
Yeah.
What? No, second sucks. Don't be second. Well, it's Talladega Nights, right? Like second is the first loser or something like that. Just kidding. Um, Multiple hosts. Yeah, Kevin, you got it right. So yeah, that's actually correct, right? So a lot of times, and this is where the trick comes in. A lot of times these are transported in E. coli. E. coli and yeast have different machinery. One's prokaryotic and one's eukaryotic. Um, yeah, a lot of times, yeah, this one could be used as different ori site. Maybe it can go into different bacteria too. Origin sites are specified for the species. So let's say maybe this one's for one kind of yeast species and this for like baker's yeast. And then you have other types of yeast species. So you're gonna have a different origin site. And then this one would probably be for bacteria, just based on the labeling. So yeah, so each origin site is based on the species you want to clone it into. So cloning it into, just as it sounds, just repeating it over and over again. When I first was learning this, I was kind of silly and I was like, oh, what does cloning mean? But that's all it is. It's just repeating it over and over again, or just inserting it into its DNA so it can get repeated. So yeah, so you have a couple origin sites there. So this is transported in E. coli. If you ordered it from Agene right now, it'd come in E. coli for you. You would start your cultures, and they would start making this based on this ori site here. But then we wanted it in yeast. But yeast doesn't recognize, the machines don't recognize this site to start replication, so you need a second origin. So whenever you're making a plasmid or whenever you're thinking about that, you want to make sure you have all the right starting points so the plasmid can actually be repeated. And make sure you really understand the kind of the thought process of what your experiment's going to look like. Starting in coli, transforming it to yeast once you get enough plasmids, so on and so forth, and having the right machinery baked into your plasmid. Now, what else makes this maybe a decent backbone or a blank backbone? Because you can see some other things here. You have this. You have a AMP R, and that just means ampicillin. Oh, actually, one question before we continue going on why it's important that you have different machinery. Can you guys talk a little bit, maybe questions? What are some differences between prokaryotes and eukaryotes? And so why is it important that we make sure we specify maybe certain machinery for prokaryotes versus eukaryotes or specific species, maybe even of bacteria or even so on and so forth, mammalian? Eukaryotes have a nucleus? Yeah, they do. Good. What else? Yeah, they have circular DNA. Yeah, no burn. Yeah, good, good. Yeah, that's pretty much, that's getting good. There's one other thing that eukaryotes do. I want you to think about the maybe even transcription translation kind of relationship in eukaryotes. Yeah, there we go. Ethan, yeah, you got central dogma. Yeah, good. Yeah, Ethan, you got it pretty good. Good, yeah, yeah, exactly. So eukaryotes even are going to have, you're going to have, post transcription and post even translation modifications done to your proteins or to your mRNA, which is going to cause a lot of different things, right? Your exons, your introns, so on and so forth. And so you also want to make sure you design something later that's going to be important for that. Another thing that's super important, and I forget who mentioned it, but good job, is prokaryotes have circular DNA. This is already kind of baked into what a prokaryote looks like. For eukaryote, this isn't the case. They have usually more, maybe it's wrapped up in chromosomes, but linearized DNA, right? And they also aren't going to do transcription and translation at the same time, while prokaryotes are. So there's a lot of things to think about when you're thinking about the species you're putting it in, so on and so forth. So our specifically is yeast. Yeast is eukaryotic. So we need to think about some of those things. It's a little more complicated than just putting it in E. coli. And so we need to be careful with our origin site. Now, one last thing. I know I'm talking about origin site a lot, but it is super important. The origin site's also important for how many times you clone. In that code, it's going to be baked how many times and how specific will it bind to the machinery in order to open up maybe the fork and have it repeat and stuff like that. And that's important, right? Because let's say we want our bread to be the greenest bread you've ever seen in your world. You're gonna need a lot of plasmids for that. You want it to replicate a bunch. Let's say we want to do a lot of experiments with it. You want more plasmids. Another thing that's important is make sure your origin sites are unique enough to be recognized. Because let's say you have 4 different plasmids, you're trying to transform this certain bacteria, maybe a ton. If your origin sites aren't unique enough, then the plasmids are going to kind of be undefined. They're only going to replicate kind of willy-nilly. That's origin sites. Very important But like I said, usually those are kind of baked in, and you can find a library for those. And so the plasmid I'm using today for us will already have the origin site in, which is really nice because a lot of people do a lot of good thinking about that. All right, let's continue. So with this, we have this AMP-R, going back to AMP-R, ampicillin resistance. Now, why do you think you want, why would you want this? Why is this important? Why would in my plasmid do I want the central dogma, so you're gonna go plasmid to RNA to protein, why do I want them to have protein that resists ampicillin? Why do I want to code for that in whatever species I have? Yeah, they're selection markers. Very good. Yeah, and exactly what Arthur said too. You want to test which one's actually transformed. So good. So, right, so let's say of E. coli, right, you're going to introduce this in. They're going to repeat it, central dogma it, and produce ampicillin resistance. Then I'm going to introduce my petri dish of them to ampicillin resistance. I can see how effective my transformation was. And that's what a selection marker is. Now, certain species, hint yeast, already has some of that resistance baked into it. So it's not a good selection marker. So we found another way, not me, but smarter people than I, have found a better way and devised a good way in order to select for these things. And one of the ways they do it is through, I always mess this up. It's like autotrophosity or something like that. I'm forgetting how to say the word exactly. It's autotrophism, I think. Yes. I'll give you the definition of what it is. So it's a species' inability to produce everything it needs in order to live. That's what it is.
It's auxotrophism?
Yeah. Thank you. And so in this case, yeast normally can produce everything. They can live on their own. However, what we have engineered strains that won't produce certain nucleic acids for themselves. So you'll have a leucine, or amino acid, sorry. You'll have a leucine that's knocked out. You have a histidine. And so what we engineer these yeast in order so they are oxotrophic. In real life, they're usually, I think protrophic is what it's called, in which they can produce the things. And so you have this thing here. This is an example of an oxotrophic selection marker. It's not as popular. The ones more popular are usually histidine or leucine. Leucine is very popular in yeast, in which will knock out the ability to create leucine, and then you'll treat it to a media. So you'll knock out the yeast ability to treat it to media. You'll introduce a plasmid to the yeast, in which it has the ability to produce that leucine, and then you'll treat it to a media that doesn't have the leucine in it. So the only ones that survive would be the ones that are able to produce leucine for themselves. That's how that experiment thought process will kind of go. And that's how you select for things that maybe already have built-in resistance. So you got to be, again, very smart about it. You got to be thinking about what kind of species am I putting it in. If I put only ampicillin resistant, my selection markers aren't going to be totally accurate. That's how that's going to work. Great. Yeah. Sorry, I need to respond to some people for the password real quick, but we'll be real quick. I realize, I guess some people did not receive the email. So let me respond. 57. Cool. I think that's it. Let me respond. Sorry, guys. Yeah. 1, 2, 3, 4, 5, 7. Cool. Want to make sure everyone can get in here. Awesome. So I'm going to pull that back up. Cool. Can you guys see my screen yet? How about now? Cool.
Ethan had a question too.
Yeah, let me check that out in 1 second. There we go. Cool. Quick. Yeah, we'll talk about that in 1 second, Ethan. That's a great question. You're preempting me a bit, and that's kind of what I was going to get at to next. No, sorry. Great question. That's an actually excellent question. Um, so the reason why we have those, and it's great, actually, great segue to talk about it now. So I found some of these backbones, and I copied them into Benchling. The way you do that, and I'll show you how to do that, because that's what you're here for. So let's say we wanted, let's do PXP518. Ooh, we'll look at it. Doesn't have the second origin site. Maybe we can add that in. Maybe we don't. But let's say this works for us now, because I have the ones I want to use. So copy paste. So Control C. You go over to Benchling here. You say, hey, I want a new DNA, import DNA sequence. I didn't download it, but I have an external file. Oh, add gene already. Command V, search for it. That looks right. Oh yeah, that is the one I said. Okay, let me import it. Bob's your uncle. There's what you coded for. Don't worry about the source thing. The source is just saying, hey, this is external. You can delete it, and I'll show you how to delete that in a second. So there we go. There we got our plasmid there. So I found some other ones I wanted to mess with. I thought this one was pretty good, but I looked at it kind of more closely. And as you're pointing out, Ethan, I looked for restriction sites. There aren't really good ones. So what are restriction sites? This is a good question for the chat. What are restriction sites used for? Why is that important? Think about how you're going to do this experiment outside of the computer. How would you actually insert a gene into a plasmid? Why do you need a restriction site in order to insert a gene into a plasmid? Yeah, exactly. There are locations that are being cut by restriction enzymes. Yeah. And so now that, no, yeah, that's awesome. Great minds think alike. So now that you've opened up, you cut this plasmid, and exactly, you've got new DNA can be added. Good. Awesome job. Yeah, perfect. So either you isolate too, yeah, if you want just one specific gene. Good. So yeah, I'm going to close my chat. So if I get questions, I apologize. So you're going to have these restriction enzymes. And if we zoom in really closely, or let's say let's tag one here. So you see here, this already marks a restriction enzyme. They're just some sort of nucleic acid sequence, only about 8 nucleic acids, 4 in length or so, which are going to cut. This one is a sticky end. You can see how it cuts. It cuts kind of weirdly, where it's going to leave things hanging off. You're going to have, it's going to look like that and that. And they call it sticky ends because these are sticky. They're going to bind to things. And this is how you would bind something to it, insert something in there. And you would add, the ends to attach it correctly. That's how you're going to insert into the plasmid. Other types they have are blunt ends, which they go straight down, but sticky ends are more common. And so you're going to have restriction enzymes in order to insert this thing in. So you want to find, when you're modeling this plasmid, you're going to want to find the restriction site that's good for you. And what do I mean good for you? Maybe a solo cut. You want to be specific. You don't want it to cut a bunch of different places and mess up the transformation or make it harder. So you want to find one maybe that just cuts one place. You want to cut in the right spot, so on and so forth. So we could add that in, or we could just find a plasmid that looks good for us. I'm looking here, and if you look here, you can get a better view of all the restriction sites and where they are. And we want it after our promoter. So again, a promoter. This is the transcription elongation factor. You want your promoter to be very specific to the type of species you're in. This elongation factor is going to be good for eukaryotes specifically. And so once it's actually inserted into the eukaryote, I can actually promote later my gene being sequenced. So that's what a promoter does. It allows you to attach the equipment for your gene to be sequenced and transcribed. Very important. You want that to happen. And so though this has a lot of good machinery, and this is the P420, you have both origin sites, you have your auxotrophic selection marker. It's not the perfect one because it's going to be hard to cut. We had to add more things in maybe. So I did some more digging. And I like this one. And I'll show you why I like this one. I'm going to move my camera over here. So if we look here, whoop, whoop, whoop. Don't worry about, sometimes I'm on a trackpad and sometimes that's difficult. So we're here. If you want to follow me, if you look at these scissors are pretty clear because it's your enzyme list, it's your restriction enzymes. And so I want to find the enzymes that kind of go here. Look at how much empty space we have. We can kind of go crazy over here. We can insert and exert things. I can move the promoter over here and feel fine about it. Ooh, I can even do this. No, we'll do over here. And it should be good, right? We'll just move the promoter over to the correct spot. So let's look at these things. So let's look at PCIL right now. So what you want to do is you want to find this enzyme. So you come down here, you click the scissors, you find the enzyme. Ooh, so two cut sites. We don't want that. We don't want two cut sites because it's not specific enough for us. We want one cut site specifically because we want to make sure that we'll be very clear where we're inserting this gene. Or when we send this off to a company, maybe they'll be very clear where they're inserting this gene because you're going to tell them what restriction enzyme to use on your plasmid. So let's do some more digging. Ooh, we can maybe try over there. Maybe we can try. Ooh, I just like the name of Eco RV. I like to be eco-friendly, maybe in my RV. And I don't know. I mean, this is actually how you would do it. Oh, two cut sites. Well, what about SWA? Ooh. Oh, there we go. That's the one. There's the money there. That's the one cut site. And that's what we want. So let's go find this on our enzyme. I don't know where I put it. I thought it was over here. Maybe it's not. But there's a lot of you can choose from. I haven't answered your question quite yet, why you have so many in one order, but you're kind of seeing why. It makes it very helpful. I don't know where it is, so we'll just find another one. This is kind of, you are seeing the magic in action of what this is going to look like. All right, SAC1's good, or SAC-L, however you want to call it. Oh, and this is a blunt one too, which is interesting. It makes you have to worry less about your sticky ends and matching them. You can just insert it at the blunt end. Maybe it makes it a little difficult, but we'll also talk about that in a second. All right, so I went SAC-L. Cool. So you double click it and it's gonna bring you over to this. So as you see here, and Ethan had this question earlier, why do you have so many restriction sites all clumped together? And what we call those are multiple cloning sites. And you can see obviously why it's good to have, because it allows for a lot, like depending on what you're trying to do, it allows you to have a great range of ability to choose your restriction enzyme, how you're gonna insert this, so on and so forth. And so we were looking very specifically for a one-cut enzyme so we can insert just a single gene, so on and so forth. Let's say you want it to be cut 6 times and get specific fragments of the plasmid, so on and so forth. Maybe you could start at this multiple cloning site and go other places. But that's why we have so many restriction enzymes around it. Because now that we introduce this to SAC-L, these are all going to be maybe more. Alrighty, so if I double click SAC-L here, it shows you kind of how it's going. I want to unselect. So I want to unselect these. Pretty easy to do. You just do that. Oh, SAC-L, I guess it's not. Oh, it's not blunt. My bad. I read it wrong. Or maybe something else was pulled up. Cool. Now we're getting cooking. So we come over here to TEF promoter. So you always need to start your gene with a TEF promoter. We're actually going to get right into this now. We're actually going to start making the plasmid to code for the GFP. And so you want a promoter, like I said, it's going to initiate transcription. So I want to copy. So I'm copying the DNA sequence, right? Include annotations and translations, see? Yes, sir. And then I want to insert it after it gets cut. Now, here's one thing, and we'll talk about this maybe in a second, but you're going to have this thing called Gibson Assembly. And this is one way, a method in which they're going to maybe put these plasmids together. We'll probably talk about this after I'm done actually designing the enzyme. Now, Gibson Assembly is going to erase this cut site. So one way you can model this is just erase this cut site now or copy over it. So I could go here and just put Command V. Another way to do it, which is to leave the cut site in and tell them. But you will tell the company this. You'll tell them like kind of what cut site you're using, and they'll be able to tell and know. So I'm going to just erase it and make sure we put it in our notes later. So if someone for me can remember that we did SAC-I or SAC-L, that'd be great. So put that in. Now we put this in. Now this is the machine doesn't know what this is, right? This is just something random to them, but we want to annotate it. So we're going to come up to here and maybe do create, or you can right click there, but we want to do an annotation for this. So this is, like I said, our TF promoter. Position, color, any good colors? Yeah, that's a good color. Save annotation. And now we have that. Now we can turn our other one off. If we remember the other color, oh, it's up here. That's ours. Yeah, we really don't really care about this one. So don't worry about that one. Cool. And so now we have our promoter. So we got our cut site. We erased the cut site because that's what's going to happen to it when we do Gibson assembly. And now we got our promoter. Don't worry about this LOXP stuff yet. We shouldn't even worry about it. There's going to be other things in your plasmid that you may not even care to code for, just around. And sometimes it's sometimes important to use operons. And if you remember Alan's thing from the first week, operons can be used to regulate gene expression. Sometimes that's important, sometimes it's not. Depending on your experiment, it's going to matter if you have those operons in there or not. So I always like to choose one, if you can see here, one with a lot of operons. Gives you nice diversity. And also you can just copy and paste, so on and so forth. So great, we got this. We got the TF1 promoter, and you're saying, well, we're practically done, huh? And I say, hold your horses. We're dealing with a eukaryotic cell again. And like we mentioned earlier, eukaryotic cells aren't just clean cut, right? They don't just go from translation to transcription or transcription to translation on one thing like prokaryotes do. They kind of go away from it. And so what you're going to want to do is have that mRNA, you're going to have your mRNA, right? The next step in this dogma of things, carry some sort of sequence to say, hey, right here, this is where I want you to start translation. Because it doesn't just translate and transcribe all at once. It needs a starter kind of codon or a starter sequence in order to do it. And so what do we call those? And oh, would you look at this? I don't actually have it pulled up, but I'll show you what it is. It's a Kozak sequence. Ooh, whoops. Kozak sequence, DNA. That's great. Here's a great website, and I'm actually going to chat it to you all now, where you can actually find this stuff and more. Be a little careful because they're not always the best. Oh yeah, that's a great question. I'll explain that. That did confuse me when I was first learning to do it. So the reason why I took out the cut site, you do need it in order to insert the gene. However, we're almost simulating the insertion. Yeah, no worries, no worries. We're trying to almost simulate the insertion of the gene. And so what we do is when we do Gibson assembly, which is a way to put these plasmids together, they eat about 30 nucleases. And so a lot of times that cut site is just going to get eliminated anyways. And so what we'll tell them is, hey, make sure you use this specific cut site and insert these genes. And we'll also send them a blank backbone. So we're going to send them this blank backbone before I edited it, PXP522, which we can get from AdGene, or we can just have copied it previously. And we'll say, this is what we want it to look like. Use this enzyme to cut it. And so they know exactly what that will look like. However, when we're done with our plasmid, this is simulating what the plasmid will look like as a finished product. And so no longer, that cut site should no longer be there because of Gibson assembly. Does that make sense? Let me know if it does not. Yeah, great. Yeah, that was really confusing when I first learned it too. Probably one of like the most confusing things about this. Cut sites. I can't even, oh no. It's now pulling up the thing over and over again. Let me try to pull up. You're gonna see how many tabs I have open in real life. Don't worry about all those tabs. Oh wait, maybe it didn't show it. So yeah, so we want some specific Kozak sequence. Like I said, Kozak sequence are a translation initiator. So once we're going from the RNA to the protein, very important, we need it for the end step of the dogma. We need these. So we want we want any kind of sequence here. So we can do this kind of sequence. We can see available, we can go on and so forth. And we can just copy paste it. I want the double stranded. This website's not the best ever. Huh, it's fine. We can find it somewhere else. So we can find the Kozak sequence DNA. This is what a lot of biology looks like too. Maybe they'll have it here. They have the mRNA, which that's actually a good example of kind of how it would translate. Cool, we'll do this. Yeah, we'll do this one. I'm going to show you something that is really cool for Benchling. So we want to insert this Kozak sequence. And you saw that I inserted the mRNA. Now, Benchling knows I'm doing DNA. So if I do that, I didn't actually click it. If I do that, it will auto correct it for us. Now, isn't that sweet? And it'll also not only do that, but do the complementary strand to it too, because again, we're dealing with DNA, double-stranded nucleic acids. So yeah, pretty cool. So again, we're going to annotate this. If anyone in chat wants to choose the color while I annotate this, go for it. No. Any colors? Any colors? Or am I just gonna, it's just Colin's choice. I'm bad. I'm very bad with aesthetic. So my plasmid's gonna look so ugly. Alan, you choose a color. Go for it.
Blue.
Well, there's 3 blues. There's like 4. Which one?
The best one.
I like this. That's like an ocean blue. You're my ocean man. Taking me by the hand. Cool. Again, press that bookmark. This will kind of help you annotate things too. Now we have this. It's a very small sequence of things, so you can barely see it. You can zoom in here and be like, oh, there's my Kozak sequence. That's so cool. All right, now we're getting to the bread and butter. We're getting to the exact thing we want. And that's the GFP. I've already pulled it up. There's a very good website too, called SnapGene that you can find a lot of this information on too. And I can put this in here for you guys if you want to go to it. Great website. You can also do look at backbone plasmids there, so on and so forth. But this gives me GFP. And a nice thing it does too is it lets me look for certain restriction enzymes. Now let's say SAC-I was here. I don't believe it is here. I thought I checked. I don't see it on one pass through. Cool. You don't want it to be there because let's say maybe some restriction enzymes stay over. Well, you just killed your gene. Well, so this will show you kind of the whole sequence. Well, this is hard to copy and paste. However, you can find this also too. This is going to have the entire sequence. Yeah. That we want. So we just copy and paste it. A lot of what this is copy and pasting. Cool. Oh, and we're going to annotate it again. All right, Lauren, this time you get to choose the color because I made Alan choose last time. This is E. coli GFP, or sorry, eukaryotic GFP. That's what the E stands for. Is Lauren still here?
I'm here.
All right. Is a sequence without introns or extrons? That's a good question. A lot of times the one I just got is just modeled for eukaryotes specifically. So they kind of already did that work for me. But if you were to try to model this protein by yourself, that is something you're going to have to carry about quite a bit. So let's say maybe you wanted introns in there, you knew kind of how they're going to get removed. You could maybe model it for that. This one's probably without introns. They probably just are coding for the exons. But GFP is a very common tag. So some of those things, it's kind of like what I was saying, where the library is built for you. So you can just kind of trust it. But that is a great question and something you're going to have to worry about when designing maybe an original plasmid. So awesome job. All right, Lauren, what color are we choosing here?
I want a dark green.
Right next to my Kozak sequence, that's a light blue. Can't believe it. All right, I'm just joking. So now we have EGFP here. Woo! This is huge. Can I get some claps? Let's go. We're doing it. All right. So what is, we need a code for one last thing here. In translation, what do you need at the end of your protein? In translation, what does your mRNA need? Chat. Woo, claps. Yeah, stop codon. You do need stop codon. I'm not smart enough to memorize them. Oh, I clicked away. All right, we're back. I'm not smart enough to memorize them, so I'm going to go to my Codon table over here. And if you don't know what this is, that's fine. We're here to learn. Codon table. Your mRNA is going to have a sequence here that code for kind of specific, amino acids. But they also do start and stopping. So we want to find the stop codon where we have TGA. There's 2 other ones, but we'll just do TGA. If anyone knows the other two, that'd be cool, but you kind of want to put them all in just to be safe. But for this one, we'll just write here. You can click here and you can say T, G, K, G, enter. There we go. Chat. How do you choose the right stop codon? Great question. So the thing, stop codon is kind of a general purpose. So in all that sequence, that TGA is going to just stop it no matter what once you insert it there. So that's a great question. But if it ever sees that TGA, it'll always stop. So the stop codons are very generic. Same with the start codons, which is super important if you want to think about the way maybe the machine of your cell works. Having generic start and stop codons will save you a lot of time and energy rather than having specific ones for maybe every protein. So you can always use that TGA, and that TGA sequence is always going to be a stop codon. Great question. All right, cool. Now we have one last thing. I'm forgetting one thing. I can't remember, but how are we going to stop transcribing? What do you need to stop transcribing? Yeah, you need the he'll be back Terminator sequence. Good. Cool. So let's do that. I already have it here because, you know, we're chilling. We're going to copy it same way with the promoter. Yeah, I want the DNA sequence, please. Just Control C. We're going to go back here. Make sure we don't cover up our stop codon or we'll never. I should have marked it too, but it's fine. Right, there we go. And this is our Terminator sequence, CYC1. And I get to choose the annotation this time. Again, this is a Terminator sequence, good for the machinery and our eukaryotic yeast cells. Probably won't be that effective in a prokaryotic cell. So again, very important. And I'm going to choose the best color you've ever seen in your life. Pink. Save annotation. Then we want to maybe turn off the gray one, just so we don't get confused. We know that's a Terminator. Cool. So that's just how I turned it off. I went up here and I did the little turn off thing. And that's pretty much it. You got it here. And we can review a little bit what we did here. This backbone would need a little more work. Some things, again, if we remember what I needed to add, I want to add a oxotrophic selection marker. So maybe my yeast, you'd have to look at what kind of yeast you're working with and what sequence it is. Maybe they're not good at making leucine. So I want a leucine oxotrophic selection marker. And I also want an origin of replication for my yeast, because right now this only works in E. coli. So those are the two other things I need to add, but we talked about them enough. And I feel comfortable, and maybe you can copy and paste it and annotate it good enough. And so what you do is then send this and your backbone off to the company, and you write, hey, we use the SAC-L or SAC-I restriction enzyme in order to cut it. And that's pretty cool. I can show you more too about like what this can do, because that's not all Benchling's used for. Yeah, that's a great question. They do. The machinery, so the transcription elongation factor is pretty general for a lot of eukaryotes. A lot of eukaryotic cells have them in, but certain species are going to detect different promoters. Some will have like very different things. Plants have different ones too. But yeah, how expensive is getting the plasmid made for you? It depends on the company. 10 to 20 cents per base pair, I think. Or base, maybe. So you're looking at like this one. I can't do math, but maybe you can. This is almost 7,000 base pairs long, $700, about 700-ish. Why don't you delete all the blank space? You could delete all the blank space here and save time and save money. I'm leaving it open right now because I would add other things into it. Is there a limit on how many plasmids you can transform into a cell? You're going to run into problems if you transform too many. So there is definitely optimizing it. And also maybe it's, you want to think about experiment design too. Remember, keep your variables low, so on and so forth. But is there a technical limit? I don't know of that. I don't know if there's like a real number you have to follow on. It's probably based on the size, on how well it transform. It also depends on the size of the plasmid. You can imagine it's really hard. Let's say you have a 30,000 long base pair plasmid. That's going to be difficult to put into an E. coli, just physically. So yeah, but you can be creative and you can add a lot of things. And this plasmid design, if you see, it kind of goes both directions because it depends on like which way you're reading it. You can really stack things on top of each other, and people who are really good at this can go crazy. Good question. Good questions. This would probably be a controlled population is like the best way to handle that. Like I said, we know the strand, we've coded the strand to not even produce leucine, so on and so forth. So we'd probably not even introduce them to wild types. But yeah, there'd be no way, let's say, for example, for them from transforming from maybe their neighbors' plasmids, not that yeast has those, but like other bacteria maybe. That's a great question. But yeah, there's, there is genetic engineering, gene drives do this too, in which you do mess with mating abilities of animals. That's how they trying to deal with the malaria and mosquitoes, is they're trying to kill mosquito populations through genetic engineering, possibly. Great question. And they want them to breed with the wild type because then they can pass on the genes. Why are some self-sufficient? Yeah, that's a great question. That's a great question. We engineered them that way. So wild type yeast are good. They can live on their own, but we engineered specific strands in order to input these selection markers in them, because we said trying to engineer them to be not resistant to certain antibiotics would be too hard. So it's easier to then make them so they're not able to produce, like, let's say, leucine. Great question. Great. All right, I'm going to show you this one last thing. So we got the Sac-I thing, and let's say I want ACE. Yeah, this one, this restriction enzyme. I don't even know what it looks like. What you can do here is you can simulate the digest, and this is super useful because it kind of, if you look over here, it will tell us how to run the experiment. It tell us how long it is, the lengths we'll have on our plasmid. It will tell us, so once we actually split it, because we're splitting it two, so we're gonna have like two linearized strings. I'm going up here, but I realized my camera's here. Tell us how long there are, tell us how long the overhangs are. Like I said, they're probably sticky. Tell us what temperatures we need to run it at, that's best optimal, so on and so forth. And so this really gives you the experiment design for you. Let's say if we had this plasmid and we wanted to add a gene in ourselves. That's super cool. Thanks, Benchling. Furthermore, this is pretty cool also. I'm gonna show you this also before I go into maybe even Gibson Assembly, because we have like time. I've been talking really fast, so thanks y'all for like dealing with that. You can create a primer for this and forward, because we're not running RT-PCR because we're not fools. It'll tell you kind of like everything you need to know about this, what temperature you need to run at. You can annotate it yourself like we did other things. We can check secondary structures too. So is this maybe actually a good kind of primer to deal with, and so on and so forth. You want this to be low because you don't want a ton of things sticking to it, and you don't want a ton of things binding. You want them to be free, and only at a certain temperature do they bind well. So, and they'll also tell you what's the optimal temperature to run at. That's pretty darn hot. Actually not that bad. It's kind of hot. What will happen sometimes, they get too hot, they'll bubble up and blow up on you, and then you'll end up with DNA all over you. It's no fun. Not that that's ever happened to me. Great. So one last thing. I know I talked about it earlier. What is Gibson Assembly? We're going to kind of move away from more specifics about it, and I'm going to show you one last feature. So if I go over here, I can go over to my little Benchling guy, and you can see here, getting started. Don't worry about that. We got Gibson Assembly protocol. So you can see, ooh, look, I can keep even protocols in here. So Gibson Assembly, I'm not going to go through this protocol with you because it can be kind of long and complicated, but this is a really good way in order to create those plasmids. It's very efficient. In one step, you can have 5 plasmids come together. In 2 steps, you can have 15 different kind of fragments come together into a plasmid. And this is why, like I said, we would model it in a certain way, because we're not just inserting it in one gene. we'd be using Gibson Assembly. And the way this does this is it creates, remember I talked about those sticky overhangs that maybe look like this, that if we just cut it would be about 5 nucleic acid long. They create it so it's like 30 nucleic acids long. You eat a bunch of ends, and so you create these super long overhangs like this that makes it really easy to bind to. You design them to bind directly to those, and so you can insert genes that way very easily. So Gibson Assembly, it's awesome. And the protocol is right here in Benchling, and all you have to do is search for it. Isn't that amazing? They said, wow, you're messing with plasmids a lot, Colin. Have the Gibson Assembly protocol on us, which is pretty sick. What else? Projects. Oh, one last thing. We want our notebook, right? We want to make sure we kind of write what we did in here. So we'll put an entry. What? Yeah, there we go. Cool. So what do we want to type here? It gives us a date and a time. So what enzyme did we use? Yeah, so we cut using SAC-L and we inserted, sorry, I'll answer this question in one sec. We inserted eukaryotic GFP. And that's what we can say. And people know what you're talking about. So yeah, so we go back under to inventory and we can look at this and we can go, once I get back over here, Plasmid, you can look at here, you can look at the linear map, so on and so forth, and you can go, get out of here, restriction enzymes. Oh, so pretty. Oh, so pretty. If you want this to look better too, this source thing, we'll eliminate it because it looks ugly. So that's what your like Plasmid will look like. That's a good question. That's a good question. A lot of times what you'll have is you would already, before you would introduce the restriction enzyme in the mix with along with the gene you want to insert in there. And so you'd hope you just have then you're running an odds game here, right? Where if you insert enough genes, it should bind enough. And that's one good way to test, like, if you include like some other things in your promoter regions, you can see how effective it is at transforming it. And you want to test your plasmids before doing it. That's also a good way on why like Gibson Assembly is really good, because it's not as easy to do that. And the way you code very specifically kind of prevents that little short overhang, which would make them easier to come back together. So yeah, good question. Great question, Arthur. So cool. So we got this, yay. Now, a fun thing, me and Lauren are going to probably use something like this in the future to actually make this bread. So stay and look out for that. We're going to use this baker's yeast and transform them to make glowing bread and other things too. I always forget the name of the banana thing. What's the name of the banana thing? Is it isoamyl acetate? Hi, I got it. Let's go. Isoamyl acetate, that's the banana flavoring in Laffy Taffy. Other things too, we thought about making it more buttery. Since we're in California, we thought about adding THC to it because we're in California. Yeah, no, exactly. That's a great question. There are different temperatures, and I can show you that. So like I said, this simulation thing helps. So let me just scroll randomly. Let's say I want to go this to this, and I run a digest. You can see the temperatures. This one's 37, 37. That's going to kind of be what restriction enzymes you want there. But like I said, there are going to be different temperatures for optimal, and this will help you simulate it. Great question. That's a great question, Emma. There's some ways to prevent that, maybe in your baking methods, but there is a good chance it will denature. There are also different proteins we can look at in modifying GFP in order to make sure that it maybe is stronger and can resist heat. So great question, and me and Lauren are thinking about it because that's one of the problems we're trying to deal with. And the one way to find out too is to try, make the bread. Another thing I wanted to mention too, and just good experiment design and kind of how this is going to work. Like I said, this is like $700 at cheapest. More likely we're looking at 800 maybe to 900. And you're like, that's pretty expensive. And unless you're like a really fancy lab, which BIOME, we are a fancy lab, I'll do say so ourselves. We're not the most strapped for cash, or we are strapped for cash. I don't know how that works. I don't know how that phrase works. Yeah, Lauren kind of answered that. Good. And so what was I saying? Oh, we want to save money as much as we can because so we don't deal with that. So one thing we'll do is we'll look at this and we'll be like, that's good design. Well, most likely, like I said, these plasmids have a lot of the same parts. And so what we'll do is just order this part here, and then us ourselves will go and be like, we have the restriction enzymes, and we'll go modify our plasmid that already has its parts and cut out things we don't want, and so on and so forth. Not as easy. It kind of sucks to do those experiments. I'm going to be frank with you, but that's life in the city. You just got to live it sometimes. You got to get your hands dirty, and yeah. So yeah, that's super exciting. Imagine being disappointed your bread isn't fluorescent green. You're already speaking my future. I'm going to be disappointed. Wait, is GFP edible? Maybe. Yes, maybe. We don't know. We'll find out. I think Lauren, no, yeah, no, not supposed to, but I had a chemistry teacher in high school, a great teacher, and she'd always tell me you can eat anything once. So what is that one thing going to be for you? Mine may be GFP bread. I don't know.
Okay, I think it's just, is it legal?
Oh, yeah, well, what, laws and things. Sometimes once not endorsed by BIOME. Sometimes laws are meant to be broken. There we go. Not endorsed by BIOME. And that law is eating GFP bread. Great. Awesome. So yeah, so great. I'm here to stick around. That's like the hour. And I'm really happy that I filled it up because I didn't think I was going to. Went really fast. If you have any questions about any of these websites, please feel free to check it out.